Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of June 2023 Shift 2. Shift 1 I have already uploaded in my channel. You can refer to my channel for the same. Here I will be discussing part B questions which were asked in Shift 2. If you are new to my channel, please do like, share and subscribe. I have also discussed old CSIR question papers also. You can refer to my channel for the same. Coming to the first question. These are part B questions. So, reduction in the frequency of heterozygous genotype with a concomitant increase in the frequency of homozygous genotype in context of random mating is due to. It is due to intense inbreeding. Inbreeding leads to an increase in homozygosity and at the expense of heterozygosity. So the answer here is option 2. Which one of the following option best represent the sequence of events leading to the phenomenon of interogression? So introgression is transfer of genetic material between species following hybridization backcrossing to the parental species and further leading to stabilization. So the best option that represent the sequence of events leading to the interogression is hybridization, backcrossing and stabilization. Answer is option 2. Then next one. In lac operon, a nonsense mutation in the gene encoding beta-galactosidase was found to interfere with the expression of downstream permease and transacetylase genes. So, there was a nonsense mutation in beta-galactosidase, or beta-galactosidase is mutated, and this mutation resulted in the non-expression of permease and transacetylase genes which are located downstream to beta-galactosidase. Which one of the following may explain this observation most appropriately? So such an effect, that is a mutation that affects transcription or translation of a part of the gene or operon downstream to the mutant site is called as polar mutation. So the answer here is option 1, polar effect of the mutation. Next question. A colorblind father has a daughter who is also colorblind and has Turner's syndrome. The genotype of the daughter is due to. Now Turner's syndrome is 45X0. So 1X has been already been provided by the father. But the mother doesn't provide the X chromosome. That is why there is Turner's syndrome. So the daughter is colorblind means she has inherited the disease from her infected father. The condition of Turner syndrome is not related to father's colorblindness. This is due to non-disjunction of chromosome during meiosis in the mother, where X chromosome failed to separate properly. So the answer here is option 3. Then next one. A cruciform structure of the chromosome during meiosis is characteristic feature of translocation. Now next one. Which one of the following is not typically a product of fermentation? So it is green tea, which is not a product of fermentation, whereas black tea, it is rich flavor is due to fermentation. Then which of the following statement is correct? None of the virulence gene of agrobacterian tumefacients are expressed constitutively. No, we have Vir, A and G which are expressed constitutively. So first statement is not correct. Integration of tDNA with the nuclear genome of a plant cell occurs only by homologous recombination. It is not by homologous recombination. It is by non-homologous recombination. So B is also not correct. Host plants do not play any role in agrobacterium mediated transfer. They play a role in agrobacterium mediated transfer of tDNA into plant cells. So third is also not correct. So what is your answer? Naturally it is option 4, which is the correct answer. Opines are source of nitrogen for agrobacterium cells. That is correct. Then next one. Which of the following statement is true? A, B and Z DNA are left-handed. No, only Z DNA is left-handed. A and B are right-handed. So A and B DNA helices are right-handed. Z DNA helix is left-handed. That itself is the correct answer. Then next one. Introns in the eukaryotic gene are found in rRNA, mRNA encoding gene but not in tRNA encoding genes. No, they are found in rRNA, tRNA and mRNA encoding genes. So the answer here is option 4. Then next one. In eukaryotic genes, DNA sequence that define gene promoter occurs. So for RNA polymerase 2, it is upstream of the transcription start site. For RNA polymerase 3, they are located inside the transcribed DNA sequence and such promoters are called as internal or intragenic promoters. Now read the options. Only in the region upstream of the transcription start sites. No. Only in the region that represent the transcribed parts of the gene. No. Only in the region downstream of the transcription termination sites. That is also not correct. Either in the region upstream of the transcription start site or within the transcribed region of the gene. That is the right answer. Then next one. To examine the in vivo co-localization pattern of two different proteins using fluorescently labeled antibodies, which one of the following combination of fluorochromes will be appropriate? So you have to see for two different fluorochromes that emit light at different wavelength. You have two different proteins and you want to know if they are found close to each other. So you need one protein of one color and labeling and the second protein of another color labeling. That is why you need two different fluorochromes that emit light at different wavelength. So as per the options, it is Alexa 488 and CY5. Alexa emits at 488 and CY5 at 647 nanometers. Next question. A plant heterozygous for a dominant trait was selfed. The progeny had 140 plants showing the dominant trait and 20 plants showing recessive trait. A researcher hypothesized that these are two genes with identical function that control the dominant trait. The researcher also proposed that the two genes are not linked. The researcher carried out chi-square test to test the hypothesis. Which of the following option is the correct chi-square value obtained by the researcher? Now he has hypothesized that two genes with identical function that control the dominant trait. That means it is the case of duplicate gene interaction and the ratio for duplicate gene is 15:1 in the F2 generation. So this is the expected ratio, that is 150:10. Over here you can see in the photo. 150:10. This is for dominant and this is for recessive ratio. The observed ratio is what? It is 140 plants showing dominant trait and 20 plants showing recessive trait. So 140:20. Now substitute these values in the chi-square formula and you will get the answer as 10.67. Now next one. The col-E1 plasmid has a low to medium copy number. However, PYQ18, which is also a col-E1 based plasmid, has high copy number because now there is a mutation in the RNA1, antisense RNA, and does not carry the ROP gene. Now what is this ROP gene? ROP is a repressor of primer, is a small dimeric protein that participates in the mechanisms that control the copy number of the plasmid of the col-E1 family. So ROP gene is not there means naturally it will be high copy number. Now it has a mutation in RNA2, that is primer for replication initiation, and does not carry the ROP gene. That is the most apt answer. So the answer here is option 2. Now which of the following statements about tata binding protein TBP is not true? It is a component of transcription factor TF2D, that is correct. TBP recognizes the tata element by inserting one of its alpha helices into the major groove of DNA. It is not alpha helix, it is beta sheet into the minor groove of DNA. So the correct answer is option 2. The TBP DNA interaction causes the DNA to bend. That's also correct. The TBP DNA interaction is governed in part by the intercalation of the side chains of phenylalanine residues of TBP between the base pair at the two ends of the tata element sequence. So that is also correct. So your correct answer, which is they have asked in the question as not true, is option 2.